Welcome everyone. Today in this video we'll be discussing about asexual reproduction. Asexual reproduction is a form of reproduction that involves the production of new organism by single parent only, that means without involvement of any gametes. It takes place by 6 different methods: fission, budding, spore formation, regeneration, fragmentation and vegetative propagation. Fission refers to the division of parent cells into two parts. This is the simplest type of reproduction and it is seen in simple single-celled organisms such as amoeba and paramecium. In amoeba, the nucleus first divides into two equal parts. This is followed by the division of the cytoplasm. Thus, two daughter amoeba grows and attain full size and then split again and again. In usual condition, amoeba divides into two parts, which is known as binary fission. But if amoeba gets favorable condition, that means enough of food and nutrition, then its nucleus could divide into multiple numbers and thus multiple daughter amoeba will produce. That is known as multiple fission. In budding, one or more new organisms grow in the form of a bulb-like projection called bud. It grows and may eventually break away from the parent organism. A little cytoplasm accumulates at one end of the cell and the bud is formed. The nucleus divides into two and one of them enters into the bud. In yeast, the buds form rapidly and create a chain of yeast cells if there is enough supply of nutrition. In hydra, bud appears on the body wall, which grows into a full hydra in a day or two and separates from the parent organism. A spore is a microscopic reproductive unit. When spore case bursts, then these spores spread into the air, then they germinate into ground and produce new plants. Bread mold that can often be seen growing on a moist stale bread grows when spores settle on the bread. In the regeneration mode of reproduction, an entire new organism can grow from some pieces or cells of the parent organism. A planaria, which is also known as flatworm, can be cut into many pieces and each pieces will grow into a complete new flatworm. In some organisms such as Spirogyra, flatworm or sea anemone, the organism breaks up into two or more fragments after maturation. These fragments grow into new individuals. Vegetative reproduction refers to the production of new plants from the parts of a mother plant, such as root, stem or leaf, without help of any reproductive organ. For example, there are several buds present on the surface of a potato called eye. A piece of potato with at least one eye on its surface is planted into the soil and develops into a new plant. In ginger, a new plant is produced by some parts of underground stem. In onion, new plants are produced from the bulbs, which is an underground stem with thick leaves. In plants like strawberry, the shoots with the buds bend towards moist land and grows into a new plant. The plants of Bryophyllum reproduce with the help of its leaves. A bryophyllum leaf contains various buds on its margins or edges. When these buds fall on moist soil, then they give rise to new plants. Several artificial methods are developed for vegetative propagation, which are commonly used to grow many plants from one plant. This is referred to as artificial propagation or artificial reproduction. Some of these methods include grafting, cutting, layering, and tissue culture. In grafting, a twig of one plant called scion is kept over the cut stem of another plant called the stalk. Then they are tied up together. The tissue of the scion and stalk combine to form a new plant. This method helps in developing a high-yielding and disease-resistant species of plant. In cutting, a healthy young branch or stem of plant having buds is cut off and planted in moist soil. The cutting develops roots and develops into a new plant. In layering, a young healthy branch is bent down towards the ground and covered with moist soil. After some time, roots develop from the covered part. This is called the layer. This branch can now be cut to form a new plant. In tissue culture, a piece of tissue is cut off from the growing tip of the plant and kept in a nutrient medium under controlled conditions. The hormones present in the nutrient medium helps in the cell division. It leads to develops tough roots and shoots. These plants are then grown in the moist soil.